Hello, everyone. Marc Estigarribia here from MSQ Ventures. We are excited and honored to continue our CEO MSQ Webinar Series with CEO Haifeng Bao of BiOneCure. We are introducing his company, himself, and also his pipeline. We're talking about third generation, new modality, 8 products, leading product in targeting TROP2, which is currently in phase 2 in US. They're going to file the IND in China this year. And we're very excited to talk about the history to date, but also what's currently going on in the trials and the differentiation and the outlook for his company and also the sector going forward from his perspective. Haifeng Bao, he's the founder and CEO of BiOneCure Therapeutics. He has more than 20 years of industry experience, broad knowledge of drug discovery and development. He's an expert in translational medicine. He's worked at Insight, at Covex, that got acquired by Pfizer, Amgen, AstraZeneca, Medi. So he's led a lot of cross-functional translational teams. He participated in numerous amount of drug development programs and more than 10 oncology new drug clinical trials. He's also, he obtained his PhD in the university, the College of Virginia Commonwealth University. Thank you, Haifeng, for attending and participating at our inaugural summer CEO MSQ Webinar Series.
Thanks, Marc, for the introduction. Yeah, I'm so excited to be here.
Yes, thank you so much. So Haifeng is calling from Zhejiang, from Hangzhou, the east part of China, tonight, and we're calling you from the U.S., New York City, where MSQ is headquartered. So I guess without further ado, maybe Haifeng, you can start with a little bit about your background. We would love to learn how you got to this point in really running BiOneCure, and also maybe a little bit of history of BiOneCure to date. We're also going to give you an opportunity to present, and then we'll answer questions from the audience. But maybe we can start with a little bit of history intro.
Sure. Yeah. So actually, I went to US for the graduate school back in 1993. So then I went to the Medical College of Virginia Commonwealth University to obtain my PhD in pharmacology. So after graduation, so I started to work for the pharmaceutical industry. And as you just mentioned, so I have worked for several different companies, and it has already been like 20 years. Mm-hmm. So back to the early stage of my career, so my research was focused on the drug target identification and validation. And around the 2004, so I started to work into the area of the translational sciences. And since then, so I worked in this area to support the anti-cancer new drug development. And so my research has been involved into the different modalities of the new drug, including like small molecule inhibitors, antibodies, of course, the antibody drug conjugates. So now it came back to the 2016, and so that time, as we know, the ADC area was actually in the downturn, and many companies thought ADC was very difficult to develop. And So since I have worked for the MedImmune, and there, so I was involved in the ADC development. So actually in the company, so we got some like the new ideas. We thought, the ADC technology should be shifted to the low potency toxin and the high DAR value instead of the very potent toxin and the low DAR ADC, which was the mainstream of the ADC development at that time. So, however, this is a kind of new at that time. And in 2016, so I left Medi and started the company BiOneCure together with my former colleagues back to the COVAX time and the Amgen times. So we founded the BiOneCure Therapeutics with the belief that we should develop a new technology, new link platforms. So that's basically the story, how and why we started the BiOneCure company.
Very, very brave to go from big pharma companies with an idea and then take it on yourself. Very entrepreneurial of you to think of that vision and actually moving it forward. How is that feeling?
It's a little bit different. So as you can see, I have been into the scientific field for such a long time. Right. So certainly, I'm familiar with all this routine work related to research, the lab work. And suddenly I changed my path to the more like the CEO role. So it's very different. So you need to deal with the fundraising and etc. So it's right. But it's different path.
Being a CEO of a company, you have multiple hats. One, running the company, the vision, surrounding yourself with very competent team to implement and execute your vision. And then you also have to raise capital and have another shareholders and board. It's a very busy but exciting opportunity, would you say?
Yes, yeah, agree.
Great. So I know so before we get to your presentation, earlier, like I think last year around April, you announced the FDA clearance for IND for BiO-106. So it's a novel TROP2 ADC for advanced solid tumors. Can you just, first of all, congratulations on that. Can you talk a little bit about that heading, that milestone achievements and where you are right now?
Yeah, so we got like the FDA's approval, and we started dosing our patients back in May last year. And so the study is still ongoing. And so far, it goes well.
Great. What is the next catalyst for on the U.S. side for these for 106 that we should expect in the 6, 9, 12 months time from now?
Yeah, so we have the plan, to expand the study into the specific tumor type or the indication, because the phase 1A or first time in human study is for the all comers. So we have done a great translational study in preclinical, and we have identified some tumor types that are particularly sensitive to the BiO-106 or TROP2 ADC. So we would like to test this molecule in these particular tumor types.
Interesting. Recently, in the second quarter of this year, you also are working together with Nanjing in biolabs. Nanjing leads biolabs to develop innovative ADCs for solid tumor treatment. Congratulations on that collaboration. Can you share a little bit about the objective and also any other elaboration you can provide?
Yeah, that company, the Nanjing Leads Bio Lab, is specialized in the antibody drug development. So they have very, very good, antibody technology, and they have discovered some antibodies for some very interesting and novel drug targets. So our collaboration is to develop antibody drug conjugates. So they provide the antibodies and use our payload linker technology. So that's a good combination, and we have done great so far. So I'm expecting, we will move these projects faster, and sometime we will have it in clinical.
Excellent, congratulations. Looking forward to following up and seeing how that develops. I guess without further ado, we could, I guess we have one more question we can ask. You are, congratulations on all the milestones and achievements in your pipeline and the geographical push in the US and also in China. Good luck with that. What is your strategic global regional partnering strategy? Is it something that you're open to? I'm sure you've been talking to MNCs for a while. What is your regional strategy, globally speaking?
Yeah, so we have developed 3 different linker payload technologies. So we have used this technology to develop different ADC products. So since ADC is very hot and many different companies want to go into this area, but they do not have their payload linker technology. So for us, we have changed our business model a little bit. So we would like to share our technologies with other companies who feels interested in. So this is the one part of the decision we have made. And the other part is that we have had the BiOne-06, which is now in clinical trial. And we have the second bispecific antibody, ADC. So many other companies feel interested in. So we are looking into the opportunities to co-develop, or we can have some kind of out licensing or the partnership to develop these 2 molecules as well.
Great. Great. Thank you so much, Haifeng. I really appreciate that. We learned a lot and looking forward to learning a little bit more. We will now go into the presentation. After you present, we'll field some questions from the audience. So thank you very much. Let's start with the presentation. Please go ahead.
Yeah, so next I'm going to talk about and introduce our BiOneCure Therapeutics, our company, next. So BiOneCure was established in 2017 with 2 locations. So one is in Maryland and the other is in Hangzhou, China. So since its inception, BiOneCure has been dedicated to the development of novel antibody-drug conjugates for the treatment of various types of cancer. So our research efforts have resulted in creation of 3 proprietary payload platforms. And based on these platforms, we have successfully developed a range of ADC products, including monoclonal, bispecific, and immune-modulating ADCs. Next. So BiOneCure has a very seasoned management team and with deep ADC expertise. Our management team comprises industrial veterans who possess expertise in diverse fields, including pharmacology, pharmaceutical chemistry, bioanalysis, clinical development, translational science, and CMC. So each team member has previously worked in ADC-related fields and brings valuable experience in our ADC new drug development. So I'm very honored or feel very happy to have the opportunity to work with this group of scientists. Next. So as we know, the success of ADC new drug development lies in the innovative payload linker technology. So from the very beginning, our company has been focused on developing our own unique payload linker platforms. To date, we have successfully developed 3 distinct platforms with different mechanisms of action. The first platform we developed is called TAM. It is a payload linker system derived from mirtazine. So TAM offers improved hydrophilicity, allowing for the creation of high DAR ADCs without causing aggregation or poor pharmacokinetics. Furthermore, TAM exhibits lower off-target toxicity as compared to SMCC DM1. So this platform has already been utilized in our first product, the BiOne06. Our second platform is centered around a topoisomerase 1 inhibitor, known as the Top1I. So this platform will be discussed in more detail in the upcoming 2 slides. And the third platform introduces a new mode of action toxin, possessing broad anti-tumor activity and featuring a more stable yet cleavable linker. But we would disclose more details in future. And this third platform will be used in some of our other projects. So these 3 platforms grant us the capability to tailor our ADC drug design based on the specific characteristics of the drug target and the associated disease. So in the subsequent slides, I will delve further into the details of the topoisomerase 1 inhibitor platform. Next. So the development strategy for the Top1I platform involves 3 areas. The first is to design and screen novel derivative molecules based on the exatecan. Then we tested various linkers to enhance and maintain the good stability and hydrophilicity. And the third part is to implement a novel strategy to connect the selected linker to the toxin molecule. So through this process, a number of payload linker candidate molecules were identified, demonstrating exceptional activities as illustrated in the figure on the right. So this candidate compound showed high potency with an IC50 of approximately 0.1 nanomolar in target positive cells, while maintaining a range of several hundred nanomolar in negative cells. So indicating the good linker stability and the broad therapeutic window for some of these candidates. So notably, some of the candidates circled in orange showed the better anti-tumor activity and the therapeutic window when compared with the benchmark Herceptin DXd molecule. Next slide. So as shown on this slide, on the top right, the payload linker molecule we finally selected for the top 1I has exhibited more potent anti-tumor activity as compared to the DXd ADCs in the xenograft tumor model. So the top 1I ADC also demonstrates the bystander killing effect, just as the DXd ADC does, as shown in the figures on the bottom. So the more characterization of the top 1I platform demonstrates the excellent tolerability and safety, along with good plasma stability and the PK profile. Its developability is also very impressive, as it can be easily conjugated with minimum aggregation, even at high DAR. Next. So now let's look at our pipeline and the exciting programs we have underway. So our pipeline currently consists of 8 programs, so each with its unique focus and potential. Our lead molecule is TROP2 ADC, the BiOne06, which is now in phase 1 clinical trial in US. And the second project in our pipeline is the innovative dual target bispecific ADC designed to target the TROP2 and the HER2. So this molecule also utilizes our in-house developed payload, the top 1I. BIO201, the candidate molecule for this project, is planned for the IND filing early next year. So additionally, we have BiOne07, a program targeting a cancer stem cell target. So we anticipate filing the IND for this program probably by the end of next year. So in our pipeline, we also have 2 ADCs, the BiOne08 and the BiOne09, that focus on drug targets related to the immune modulation. So we also have the project that BiOne10, which targets a novel drug target, so expressed in the various solid tumors. So it could be a first-in-class ADC with broad anti-tumor activity. So more interestingly is the other 2 projects, the BiOne11 and the BiOne12. So these 2 projects are not conventional ADC programs. So these two molecules employ a unique approach by selectively killing some normal cells in the tumor microenvironment to enhance the anti-tumor activities of other chemotherapy or immunotherapy drugs. So from this, our pipeline, basically it just represents a diverse range of the programs, so reflecting our commitment to advancing novel ADC therapies. Yeah, next, please. So now let's delve into the details of BiOne-06, the anti-TROP2 ADC. So as we know, TROP2 is overexpressed in various solid tumors, presents an excellent target for achieving broad anti-tumor activity and tapping into the significant market potential. However, its expression in certain normal tissues leads to the potential risk of on-target toxicity. So when we designed the BiOne-06, we select a non-cleavable link. So this design choice aims to minimize on-target toxicity to avoid the bystander killing effect from the TROP2 expressing normal cells to the surrounding other cells. So by employing this linker along with a high DAR value of 6, we prioritize safety while still maintaining the potent anti-tumor activity. So additionally, BiOne-06 utilizes the TAM, a microtubule inhibitor, as its payload. So as we know, other TROP2 ADCs, such as the Trodelvy, Immu-132, DS-1062, and SKB-264. So they all employ the top 1 inhibitors. So BiOne-06, by using the microtubule inhibitors, set it apart from the other TROP2 ADC. Next. So Trodelvy, or the Immu-132, is the TROP2 ADC currently approved for the treatment of triple-negative breast cancer and the metastatic urothelial carcinoma. The active component of Immu-132 is the SN38, which is derived from irinotecan. So in these clinical studies using xenograft tumor models, we compared the potency of BiOne-06 and the Immu-132. So as shown on the left, so BiOne-06 has demonstrated greater potency compared to the Immu-132, even when it was administered at higher doses and more frequent dosing. So in order to identify and differentiate BiOne-06 from other TROP2 ADC, so we have studied in over 40 patient-derived xenograft models, the PDX models, to see if we can identify any specific tumor types that is sensitive to the BiOne-06, but not that sensitive to Immu-132. So it has shown high effectiveness in several types of the GI tumors that do not respond well to Immu-132. So this suggests that BiOne-06 may have the potential to treat the tumor types like some GI tumors that are not sensitive to the top 1 inhibitor-based ADCs. Interestingly, in our phase 1 clinical trial, a breast cancer patient who had previously treated with various therapies, including Immu-132, the TROP2 ADC, as well as the DS-8201, the HER2. This patient failed in this treatment, but exhibited notable tumor reduction following treatment with BiOne-06. So this finding actually indicates the potential of BiOne-06 for treating patients who do not respond to immune 132 or other topoisomerase 1 inhibitor-based ADCs. Next slide. So in summary, the BiOne is an antibody-drug conjugate that targets TROP2 and utilizes the TAM, a novel microtubule inhibitor developed in-house as its payload. So BiOne has demonstrated potent anti-tumor activities with great safety, good plasma stability, and PK profile. And it has a favorable developability. So BiOne is now in phase 1 clinical trial with a differential development plan. Next. So now let's switch the gear to our second molecule, the BiOne, the dual target bispecific ADC. So we know the HER2 has demonstrated remarkable success. So the question has since been asked, how to surpass HER2's performance? So when we take a close examination of HER2's approach, so we will find HER2 has been granted for a specific subpopulation within each indication. So this subpopulation consists of either HER2 highly expressed or mutated patients. And these patients only account for a small fraction of the total patient population. So if we can expand the eligible subpopulation for a new ADC drug, so this ADC may have the potential to outperform HER2 by reaching a larger number of patients. So that's why we designed this BiOne molecule. It targets HER2 and the TROP2. So why we choose TROP2? So because TROP2 exhibits the high expression level in very solid tumors and appears to be suitable companion target to HER2 in terms of the prevalence and the expression levels on tumor cells. So for this molecule, the eligible patient population is supposed to encompass the patients who are either HER2 positive, TROP2 positive, or a combination thereof. So in terms of the design of the BiOne, so the antibody of this molecule consists of TROP2 IgG1 and a single chain HER2. The HER2 arm possesses the same binding affinity as Herceptin. So BiOne is conjugated with the top1I payload. So by targeting both HER2 and TROP2, BiOne can potentially expand the eligible patient population beyond the limitations seen with the HER2, this providing the broader coverage and reaching a larger number of patients. So just as we show on the right of the slide. Next. So here is the internalization profile of BiOne. So BiOne exhibited rapid internalization in TROP2 positive cells, consistent with the known behavior of TROP2 as a fast internalizing antigen. Notably, in N87 cells that express both HER2 and TROP2, so BiOne demonstrated enhanced internalization compared to its parent antibodies, as shown on the figure in the figure on the right. So suggesting the improved cellular uptake by targeting two antigens. Next. So on this slide, we can see BiO201 demonstrated potent dose-dependent anti-tumor activities across a range of tumor models. It is also effective in the HER2 negative, the MD-MB468 tumor. The TROP2 positive tumor model. So in contrast, the Herceptin ADC did not exhibit significant anti-tumor activity in this specific model. So this finding actually support the a broad anti-tumor activity of BiO201 as compared with the monoclonal antibody ADC. Next. So on this study, we compared the potency of BiO201 with Herceptin DXd. So as shown here, so BiO201 exhibited comparable anti-tumor activity to the Herceptin DXd in tumors expressing different levels of HER2. Next slide. So in summary, so BiO201 is a novel bispecific ADC that targets HER2 and TROP2, utilizing a new TOP1 inhibitor developed in-house as its payload. So BiO201 has kept a similar potency to the Herceptin DXd in HER2 positive tumors and demonstrated broader anti-tumor activity in those tumors of HER2 negative but TROP2 positive. So BiO201 also shows great safety, good plasma stability, good PK profile, and favorable developability. Next. Yeah, so finally, I would like to thank you for watching this presentation.
Great. Thank you very much. Haifeng, we learned a lot. Congratulations on all those milestone achievements. We're looking forward to great things from BiOneCure. So we've been gathering some questions from the audience. So we have the next part of the MSQ CEO Webinar Series. So let's start with the questions, Haifeng. Can you tell us, here's one, can you tell us more about BiO106 and how they differ from other TROP2 targeted ADCs currently in development and on the market? So I think you addressed a lot of the differentiation, but maybe you can just from a high level.
Yeah. So first of all, when we designed this molecule, we employ the non-cleavable linker to avoid or minimize the bystander killing effect. So because we know TROP2 is expressed in certain normal tissues, and the on-target toxicity is always the concern for the TROP2 ADC. So by using the non-cleavable linker, so we think we can minimize the on-target toxicity. So this is the first thing. The second, so for this molecule, we use the term microtubule inhibitor. So for the other TROP2 ADC currently in clinical trial, such as the DS1062, the Immu132, and other molecules. So they all use the tubular topoisomerase 1 inhibitor as its payload. So because the MOA of the toxin are different, so we expect BiO106 may be sensitive to some tumors that may not respond well to the topoisomerase 1 inhibitor ADC. So I think this is the major difference of, or like the unique, part of the BiOne06.
Great. The next question, we have seen early results demonstrating efficacy as a monotherapy, but what is the potential for combination therapy for more advanced stage or treatment resistant tumors?
Yeah, we haven't, started the combo study yet. However, in the preclinical study, so we have done some experiment. For example, the BiOne06 induced the immunogenic cell death. So that, may provide the evidence we can combine the BiOne06 with some like the IO therapy, like the PD1 or PD-L1 inhibitors.
Mm-hmm. Great. We have a few more. So one critical factor to consider is the bystander effect and effect on healthy tissue margins. What has been BiOne06 effect on tissue surrounding the solid tumors?
So we have done some studies. So because we use the non-cleavable linker, so the after the drug molecule is internalized in the lysosome, the metabolite carries lysine. So these metabolites cannot go through the cell membrane easily. So we have tested in the in cell lines. So we do not see the bystander killing effect. So that's why we assume in human the bystander killing effect or the damage to the surrounding normal tissues will be minimized for this molecule.
Great. Next question from the audience. When considering therapeutic windows, what is the typical response rate to high doses of BiOne06? And how long is this response maintained? Just I guess a follow-on to that. Any potential for intratumor administration injection?
Yeah, for the first part of the question, so we haven't got that part yet. So I do not have the answer yet. Okay. And for the second part of your question, so I don't think we are going to do any like locally administration for this molecule.
Okay, great. Another one. How does BiOneCure's plan to move forward? So what's your plan to move forward with other assets? Are there any upcoming? I know you spoke about your pipeline. Are there any upcoming clinical trials or milestones we can expect to see in the near future? Let's say the second half of this year.
Yeah, based on our plan. So we will file IND for the BiO201, the bispecific antibody, maybe early next year. And so then we may file IND for the BiOne07 ADC that targets the cancer stem cell in the probably by the end of next year.
Exciting. What potential indications or patient populations are you most excited about BiOne06 and why?
Yeah, so we actually we have done a very good translational study. So we actually tested the BiOne06 in like in the PDX model. Actually, like from the over 40 PDX models, we want to see which tumor type may be sensitive to our molecule. And we did observe the BiOne is sensitive to the tumor types like the triple negative breast cancer, non-small cell lung cancer, and the blood cancers, which are also very sensitive to the other TROP2 ADCs. Interesting thing is that we also observed BiOne had very good potency in some GI tumors, which do not respond well to the like Immune-132. So actually, so we are very interested in to take this molecule into the GI tumor types.
Great, thank you. I think we have maybe 3 or 4 more. What safety measures has BiOneCure put in place to ensure the safety of patients in clinical trials? What are the metrics that are being analyzed over the long term to ensure an adequate response to 106?
Yeah, so we basically followed the regular design, for this molecule, either like for the preclinical safety evaluation as well as in clinical. So, this molecule is like we expect it may have like some toxicity associated with the metastatic type of payload, and maybe also some on-target toxicity associated with the TROP2 expression.
Great. Almost. So we have a few more. Could you speak to the collaboration BiOneCure has established with Nanjing Leads Biolabs? I guess specifically, what should we expect in the short term, in the second half of this year, going to next year? What are the big milestones in terms of the progress to date so far?
Actually, we start these programs at the very early stage, and so they have the antibodies, and we have the payload technologies. So first we will put them together, and as a potential, ADC compound. So we will do all these preclinical assessments, including the pharmacology and later the PK, plasma stability, as well as the safety assessment. So far, this collaboration goes very well, and I think it's on the right track.
Great, thank you. Good luck. Looking forward to hearing more progress there in terms of your accomplishments. We just have 2 more questions. So next one, are there any constraints to scaling up manufacturing of this asset and ensuring the quality and the reproductive ability among batches of drugs? Excuse me.
Yeah, so far, the BiOne, we haven't seen any issue, for the CMC or the productivity and manufacturing. And actually, we had this molecule that we have the CDMO. We have asked the CDMO to do the GMP production. So the technology or molecule was very solid, and we haven't encountered any issues yet. So I do not think, or do not expect, so there are any difficulty to scale up.
Great. We have 1 more question, and then the webinar will conclude. So here we go. So as BiOne targets TROP2 in several peripheral indications, such as breast, kidney, lung, is there capacity to cross the blood-brain barrier should cancer spread?
That's a good question. I don't know. Actually, I do not have the answer. Although I heard that some like TROP2 ADC is effective in reducing or like reducing the metastasis in brain. But for our molecule, we do not have any data yet. I don't know. We'll see.
Great. So I think that concludes our CEO webinar series. We want to thank you, Haifeng Bao, CEO of BiOneCure, to present your company and to share your knowledge. We learned a lot today. We're very excited to hear more and to further discuss your progress in the future. Good luck.
Yeah, thank you, Marc, for having me here. Thank you.
Of course, our pleasure. And that concludes our webinar series today with BiOneCure and Haifeng Bao, the CEO. Have a great day. Have a great night.
Okay, good night. Bye-bye.